Hi everyone. For today's 3-minute paper, I will highlight work recently published by scientists at Google, Triple Bar, and the University of Cambridge working to explore the differences between directed evolution, machine learning guided directed evolution, and even zero-shot learning. While each of these protein engineering approaches has been demonstrated in the literature, the authors argued that there isn't conclusive evidence whether machine learning guided directed evolution is definitively better than classical directed evolution across multiple design cycles and improving protein function. To address this gap, researchers explored the differences between these approaches in the context of engineering novel nucleases that can be potentially used to prevent biofilm formation. Biofilms are a complex web of microorganisms and their associated proteins, sugars, and nucleic acids that protect and support the growth of potentially harmful microbes, thus impeding wound healing in clinical contexts. Nuclease B, specifically the endonuclease isolated from the microbacillus licheniformis, has been shown to cleave the extracellular web of DNA, effectively breaking down biofilms. However, this particular nuclease is most active in its native alkaline environment, which is not conducive to practical clinical applications in neutral pH environments. So this project's overall guiding design goal was to engineer a novel protein with higher nuclease activities at neutral pH than the wild-type nuclease activity at its native alkaline pH. As this paper's title suggests and highlighted here on the right with this animation, one highlight of this work is the implementation of a custom microfluidics chip to sort through these libraries. And the specific fluorescent readout they're sorting for is made possible by the cleavage of a 3' fluorescent quencher away from the 5' end of an oligo conjugated to the fluorescent AF48 dye. However, it is important to note that these ultra-high throughput screens still need to be validated with higher resolution but lower throughput assays. So now, with the limited time remaining, I'm going to briefly survey the different engineering approaches implemented in this paper. Across all of these engineering approaches, except for the zero-shot case, the design cycle started with an initial random library generated by error-prone PCR, which was screened for nuclease function. Then, in the case of machine-guided directed evolution, named teleprot, either convolutional neural networks or variational autoencoders were trained to identify a population of variants to screen for the next round of design. It is important to note that authors compared several different library design methods with different combinations of training data sources, the details of which I don't have time to cover in this video. As a non-ML computational comparison to the CNNs and VAEs, hit recombination, or HR, was used in which mutations from the top variants from each design rounds were randomly combined and sampled. Interestingly, this genotype-phenotype data acquired from the HR design campaigns were used to train models of certain design methods for the ML-guided directed evolution campaign. Finally, these two engineering approaches were compared against a classical directed evolution campaign, which generated new libraries to screen with gene shuffling and error-prone PCR. So I'm running a bit over time, so briefly. As expected, each successive design round resulted in increased nuclease activity. However, the machine learning guided approaches resulted in significantly higher activities than the directed evolution campaign at pH 7. The activity of the wild-type nuclease in its native pH 9 is denoted here with this dotted line. For the cases of ML4, researchers explored the capabilities of ML-guided methods to improve the diversity of the screening libraries, with ML4 campaign having significantly more mutations with improved activities compared to the HR campaign. One intriguing highlight I'd like to close out with is our zero-shot generation demonstration. Sampling the latent space of a VAE trained on nuclease B homologs, the authors found that, compared to error-prone PCR, the hit rate of the zero-shot samples was higher for finding both enzymes with non-zero activity and even enzymes with increased activity, suggesting that zero-shot generation may be a better starting point for directed evolution campaigns. There's a lot more I didn't cover, from the discussions on ML's ability to infer epistatic relationships to the issues encountered with synthesizing these libraries, so I hope you give this paper a read. Otherwise, thanks for watching this video.